Good evening, everybody. Welcome to the webinar on the chemical modification of wood. Today. So actually here, before going for the chemical modification of wood, it is first we have to understand what is the chemistry of wood. In the chemistry of wood, especially we are concentrating on cell wall. In the cell wall, majorly contains three major chemical components. These three chemical components are cellulose, semicellulose, and lignin. Cellulose and hemicellulose both are called holocellulose. The content of the cellulose and hemicellulose is different in the softwood as well as hardwood plant species. In the softwood species, the cellulose content is 33 to the 42%. Hemicellulose content is 20 to 40%. Lignin content is 27 to 32%. And other extranaceous or extractives present in the softwood species is 2 to 3.5%. The extractives that are present are the organic compounds. These organic compounds are tannins, flavonoids, alkaloids, resins, ash content also present. In the hardwood species contains 38 to 51% of cellulose, 70 to 38% of hemicellulose, and 21 to 31% of lignin. And other extractives are present in the hardwood species is same like softwood species. They are resins, tannins, flavonoids, alkaloids, etc. Organic compounds are present. The amount of lignin and hemicellulose, cellulose, and extractives in the herbaceous plants. That means in this type of the plants, there is no developed stem is present. In those plants, the observed content of lignin, hemicellulose, cellulose, and other are 0 to 40%, 20 to 50%, 25 to 95%, and 4 to 9% of extractives are present. So after that, the main thing we have to understand from the wood chemistry is the structure of cellulose. The cellulose is the most abundant organic compound on earth, and the chemical formula of cellulose is C6H12O5 with N units. Here N value is around 10,000 to 15,000 is present. So this number of units are present in the cellulose structure. The amount of cellulose present in cotton is 90%. Amount of cellulose present in the wood is 40 to 45%. And dried hemp is 57% respectively. The major thing in the cellulose structure is the cellulose is a unbranched and non-cross-linked natural polymer. The cellulose is made with beta glucose units. That means the cellulose is a polymer containing monomers as beta glucose units. The beta glucose units are bonded together by beta 1,4 glycosidic bonds. The glycosidic bond is the oxygen linkage present between beta glucose residues. In the structure of glucose, every alternate glucose residue molecule in cellulose chain is inverted The inverted arrangements of glucose residue are responsible in making long and straight chains. These cellulosic chains are arranged parallel to each other to form microfibrils. It is actually structure of cellulose. In the structure of cellulose, we observe the beta glucose units. What all are showing here are the beta glucose units. These beta glucose units, like 4 beta glucose units, are represented here, and the 2 are represented with the N value, that is also called cellobiose. These are having inverted configuration. In the structure of cellulose, we may observe the inverted configuration, that is, in the first unit, the hemiacetal link is present in the above, whereas in the second unit, the hemiacetal linkage is present downside. And these 2 glucose units are connected by glycosidic bond. So here N represents value 10,000 to 15,000 is present. That is, it is very highly polymeric nature. Cellulose having highly polymeric nature is present for the cellulose. It is the structural representation of the cellulose polymer. Actually here what the represented is the chair conformation of beta glucose units. In before, here the rings that are present, these are also glucose units, but these are called glucopyranose units. 4 are represented there. All these are the beta glucose pyranose units. And these beta glucose pyranose units are represented in the form of chair structure. Chair structure is most stable conformation compared to this ring structure. Actually here the 4 beta glucose units are present. These 4 glucose units are present in the pyranose structures. These are also called glucopyranose units. And in this structure, the beta glucose units are present in the form of chair structure. The chair conformation is highly stable conformation according to the chemistry. So this is more preferable conformation of the glucose compared to the previous configuration. It is the general configuration only, but here the represented cellulose structure is the realistic one compared to this pyranose ring structure. This is also pyranose ring structure, but the conformation is realistic. Here the cellulose structure in which we found the intermolecular hydrogen bonding also. Intermolecular hydrogen bonding is formed in between oxygen and hydrogen atoms. That means oxygen is the highly electronegative element. This forms most stable intermolecular hydrogen bonding. So here the 4 layers are present. In the 4 layers, we may found the intermolecular hydrogen bonding. That means the crystallinity in the case of cellulose is more. The more crystallinity is found. Crystalline structure means there is a certain direction is present and the regularity of molecules is present. So all the cellulose molecules, we may found that they are arranged in regular fashion and giving more crystallinity. And cellulose is not only the crystalline molecule, which is also a amorphous in nature. So most of the part of cellulose is crystalline compared to amorphous part of cellulose. Amorphous means there is no regular arrangement of these beta glucose units in the chains of cellulose. So in the structure of cellulose, this is the most suitable and comfortable and realistic conformation of the cellulose structure, and it is a crystalline form. And amorphous form is irregular in nature. That amorphous structure of the cellulose is not represented here, but it is the crystalline part of cellulose. And the cellulose which is present in the plants is the alpha cellulose. That means cellulose 1. Next, structure of hemicellulose. Hemicellulose is another branched heteropolysaccharide which is made up by pentose and hexose sugars. That means here the monosaccharides that are present in the hemicellulose are pentose sugars and hexose sugars. Pentose means pentoses contains 5 carbons, hexose sugars contains 6 carbon atoms. In their structure, both are the aldehydes only. Pentose is the aldehyde molecule, hexose is also aldehyde molecule. Both pentose contains 5 carbon atoms and hexose contains 6 carbon atoms respectively. Pentose sugars includes xylose and hexose sugars, xylose and arabinose, and hexose sugars are mannose, galactose, glucose, and deoxy sugars like ramenose. Next to glucouronic acid. All these are the hexose sugars only. That means all these are contains 6 carbon atoms except xylose and arabinose. Xylose, arabinose, both are contains 5 carbon atoms and they are called pentoses. Hemicellulose actually contains pentose and hexose monosaccharide residues. These are bonded by beta 1,4 linked backbones. So the glycosidic linkage present in the hemicellulose part backbone is the beta 1,4 glycosidic bonds. Here the monomeric units are represented in the hemicellulose. Glucose, first molecule glucose, galactose, xylose, arabinose, glucouronic acid. Glucose is a hexose molecule and it is the most stable conformation of the glucose. That is, the molecule is present in chair conformation. All these molecules, either pentose or hexose sugars, are present in the chair conformation. But actually here arabinose is represented in the boat conformation. Of course, compared to boat conformation, the chair representation is the most suitable and stable conformation. So glucose, galactose, mannose, and glucouronic acid, all these contains 6 carbon atoms, whereas xylose and arabinose, the down represented or xylose and arabinose, both are contains 5 carbon atoms only. If we carefully observe, we may find that only 5 carbon atoms in the xylose and arabinose, but whereas the rest of our 4 molecules with 6 carbon atoms are present. And here oxygen linkage is in the chair conformation is the hemiacetal bond, and all hydroxyl groups are the major things that are found in the either in the case of cellulose monomerics or hemicellulose monomeric units. So hydroxyl content is the major either in the case of cellulose or hemicellulose. Here, based on the structural difference like backbone linkages and side routes, as well as by considering other factors as well as the distributions, hemicellulose classified as 4 parts: Xylons, Xylons, mixed linkage, beta glucons, Xyloglucons. So these are the 4 parts are observed in the hemicellulose. Xylon. So Xylon is the dominant hemicellulose in the hardwood and non-woody biomass also, like any small plants, cotton, as well as beetroot. So in this case are considered to be non-woody biomass. It's a deconstruction to monomer. That means we are doing the hydrolysis. Hemicellulose is easily hydrolyzed compared to cellulose. Cellulose is not hydrolyzed easily because cellulose is a crystalline part. So due to presence of highly intermolecular hydrogen bonding construction in the cellulose deconstruction is not possible easily, but whereas we can we can deconstruct easily the hemocellulose by the dilute acids as well as alkalis. So that means we are making the polymer into the monomeric units by addition of acids. So here either enzyme hydrolysis or by chemical hydrolysis, we can make the Xylon polymer into monomeric units. Here we are observing the structure of Xylon. So Xylon is a heteropolymeric substrate. Heteropolymer means which contains the different types of monomeric units, not only one. So different types of monomers are present in the Xylon polymeric material. So Xylon is the major part of hemicellulose. Xylon contains the Xylon monomeric units. These Xylon monomeric units are Xylon polymer contains xylose residues. That means xylose molecules are pentose molecules, and all these xylose residues are connected by beta 1,4 linkages. That means the glycosidic bond is formed in between xylose molecules. Here in this structure represents the structure of Xylon. Here the backbone are present in the black color. So the backbone present in the black color shows all the xylose molecules. That means they are the xylose residues. They are also called xylopyranose units because we can represent the xylose molecule by taking the organic molecule pyranose into the consideration. So all these xylose molecules are pentose molecules, and we find the black color in which 5 xylose residues are present, with each molecule contains 5 carbon atoms, and all these molecules are bonded together by glycosidic bond. That means any 2 xylose residues are connected by glycosidic bond. That means the oxygen linkage is found in between 2 glucose units. So here Xylon on hydrolysis produces all these units. That means if you do hydrolysis by using dilute acids, we can get xylopyranose units, acetic acid, ferulic acid, glucouronic acid, arabinofuranose. All these structures are present. So here we can observe the backbone of the Xylon is made with beta 1,4 glycosidic bonds, and the branches here in the structure we may found that the branches are present. That means the black color backbone of the xylose residues are connected to different monomers. So all those represented are the monomers. Above in the red color that represents glucouronic acid. Glucouronic acid is connected to the oxygen linkage, and acetate group also connected in the downside. Next with the green color, arabinofuranose is present. That means L-arabinofuranose. That means the conformation is the different. That means actually deconfiguration is mostly found, but whereas here arabinose is the pentose sugar that is found with the L configuration. So that L-arabinofuranose is also made with 5 carbon atoms. Again, this arabinose is bonded to ferulic acid. So the ferulic acid is the present in the form of esteric representation, so which is derived from the ferulic acid. So that is found in the blue color. Again, here where the linkages are present in the with the backbone or backbone branched with acetyl group, arabinofuranose. Next, 4-oxymethyl glucouronyl groups. In addition, Xylon may be cross-linked to lignin by aromatic esters. Here in this structure, it is the aromatic ester, ferulic acid. So that aromatic ester is the component of lignin. So Xylon is the material of hemicellulose polymer, which is again bonded to lignin also. So individually, any component is present. So that means hemicellulose lignin components are not separated. They are bound. They are bound together by the bonds that like that esteric linkage here in the Xylon. The esteric linkage is found in between lignin material with the hemicellulosic material, that is Xylon. In order of efficiency, depolymerize Xylon to the component monosaccharides, a mixture of different enzymatic functionalities are required, including, so all these are the enzymes what we are representing. So with these type of enzymes, we can make a depolymerization. So after the depolymerization, we may get the monomeric units. So after the formation of the monomeric units, we can understand that what are the monomers that are present in the Xylon, which is a part of hemicellulose. Next, second part which is present in the hemicellulose is the mannans. So the mannan type of hemicellulose can be classified into two types based on their chain difference. They are the galactomannans and the glucomannans. Actually, mannans are also present, but the mannans are mostly found in the grass, and the galactomannans and glucomannans are majorly found in the wood. So galactomannans have beta 1,4 linked mannopyranose residues in the linear chains. Okay, so here, so diagrammatic representation is not there, but actually here galactomannans contains mannopyranose units. So these mannopyranose units are found in the form of the linear chains and that are bonded to the galactose, which is a hexose sugar. And glucomannans consist of both beta 1,4 linked D mannopyranose and beta 1,4 linked D glucopyranose residues in the main chains. So as for the side chains, D-galactopyranose residues tend to be 6 linked to both types as the single chains with various amounts. So here the figure is not given, sorry. So actually here in the sixth position of the either galactomannans or glucomannans, the galactose unit is present. So that is in the case of mannans. Next, mixed linkage beta glucose. So in the mixed linkage beta glucons, the conformation of the mixed linkage glucon chains usually contains blocks of beta 1,4 D glucopyranose separated by single beta 1,3 beta glucopyranose units. So that means in the mixed beta glucons, only glucose units are present, but the linkage is different. So alternative units are bound together in the first linkage with 1,4, but whereas the second glycosidic bond with the 1,3 glycosidic linkage. The population of beta 1,4, beta 1,3 are about 70 and 30%. These glucons primarily consist of cellotriazyl and cellotriazyl segments in the random order. Actually, this cellotriazyl is a 4-carbon monomeric unit. So actually, 3 carbon atoms on which the monosaccharide units are started. So here we found that in the case of hemicellulose, we found that this aldoses are present in the form of 4 carbon units. That means tetramer, tetrose. That is also called tetrose. That means cellotriazyl is the tetrose. 4 carbon atom unit is present. 5 carbon atoms containing monomeric units are xylose and arabinose. 6th carbon atom containing monomeric units are mannose, glucose, galactose, glucouronic acid. All these are the 6 carbon atom residues. Next one is the xyloglucons. Xyloglucons have a backbone similar to cellulose consisting of beta 1,4 linked D-glucons substituted with xylose with alpha D xylopyranose residues at position 6. So xyloglucons. So this is the another important part of hemicellulose. So the backbone present in the xyloglucons is the D-glucons. That means D-glucose. D-glucose is substituted